For this lecture, we will discuss heredity. This is a picture of Gregor Mendel, who in the 1800s discovered through experimentation that traits are determined by elements that are passed from parents to offspring. In the early 1900s, this element was christened the gene. Genes are the smallest units of information in chromosomes. They contain the instructions for producing certain cellular products that result in certain traits. A locus is the physical location of the gene on the chromosome, and alleles are different forms of a particular gene. They occupy the same loci on homologous chromosomes. If we want to refer to all the genes an organism possesses, we use the term genome. Now this term also includes nucleotide sequences that do not code for cellular products. For example, proteins bind to specific nucleotide sequences in DNA to mark where the origins of replication will begin during DNA synthesis. These nucleotide sequences do not contain the recipe to make any product, but merely serve as an attachment site. The human genome consists of about 23,000 genes found on 23 pairs of chromosomes. Here we have a pair of chromosomes. These chromosomes are found in the genome of a pea plant, the organism that Mendel used to discover genes. As the chromosomes have the same sequence of genes, they are homologous. This locus on the chromosome is where we find the gene for flower color. And we have two different types of flower color genes that can be found at this locus, the purple flower allele or the white flower allele. So how did Mendel figure out that genes were passed from parent to offspring? Through experimentation, of course. Mendel chose the pea plant as his model organism because of its reproductive abilities. It can self-pollinate because the flowers contain both the female and male reproductive organs. The male organ produces pollen, which makes sperm, and the female organ produces eggs. However, pollen can be carried by the wind, so it's difficult to determine who the father plant might be. But Mendel could selectively cross-pollinate the pea plants. He removed the male organs from a flower and then physically transferred the pollen from one plant to the female organ of another with a brush. Then he could be sure of who the father plant was in a cross. So Mendel selected pea plants with certain characteristics to serve as the parental or pea generation. Then he selectively crossbred these parents. The female reproductive structure produced seeds and could be planted to produce the next generation. Yes, peas are seeds, and seeds contain plant embryos, so you can think of seeds as plant babies. So Mendel would breed these pea generation plants. He made sure they were true breeding. That means if crossed with another plant with the same trait, all the offspring would have that trait. For example, a plant with white flowers is crossed with another white-flowered plant. Only white-flowered offspring are produced. The pea generation plant would be considered true breeding. Now when he crossed purple-flowered plants with white-flowered plants, the offspring were all purple-flowered. He called this generation the F1 generation, and when he crossed members of the F1 generation, about 3/4 of the offspring had purple flowers, while 1/4 had white flowers. This next generation he called the F2 generation. Now, how did the white flower trait disappear in the F1 generation and reappear in the F2 generation? Let's learn some more terminology first. Gene expression results in the production of a cellular product, typically a protein. The protein manifests as a trait. When we use the term character in genetics, we mean a feature that the offspring inherit from their parents in the form of genes. Flower color would be an example. But the term trait is more specific. Traits are variations of characters, white or purple flowers. A dominant allele is one that expresses itself over the second type of allele, the recessive allele. In our initial examples, the dominant allele will be written as a capital letter. An uppercase P is used for the purple flower allele. The recessive allele is only expressed if both copies in the gene pair are recessive. We will denote recessive alleles with a lowercase letter. A lowercase p is used for the white flower allele. If both the genes in the pair are the same type of allele, they are called homozygous. If both alleles are for the purple flowers, we would say the plant is homozygous dominant for the flower color. If both alleles are for the white flowers, we would say the plant is homozygous recessive. If the alleles differ, the individual is heterozygous for that character. If the plant has a purple flower allele and a white flower allele, it is heterozygous for flower color. The actual alleles the organism possesses for the character is the genotype. A plant with 2 dominant alleles for flower color is homozygous dominant. A plant with 2 recessive alleles is homozygous recessive, and a plant with 1 of each is heterozygous. The physical expression of the gene is called the phenotype. A plant that is homozygous dominant for flower color produces purple flowers. A plant that is homozygous recessive produces white flowers, and a plant that is heterozygous produces purple flowers, since the purple allele expresses itself over the white allele. The results of Mendel's work are very interesting. If we look at the characteristics he examined, we can see a definite pattern. In seed shape and color, pod shape and color, flower shape and position, and stem length, all the F2 generations showed a 3-to-1 ratio of phenotypes. That means that the dominant phenotype expressed itself 3/4 or 75% of the time, while the recessive phenotype expressed itself 1/4 or 25% of the time. Let's see how these crosses can be calculated. Mendel was looking at monohybrid crosses in which only one characteristic was being examined. First, we have to determine what type of gametes the P generation can produce, and to do that, we have to know the genotypes of the parents. Since the plants are true breeding, the purple-flowered one must be homozygous dominant, and the white must be homozygous recessive. Through meiosis, each can produce haploid gametes with 1 homologous chromosome from the pair. So the homozygous dominant parent can only produce gametes with the dominant allele, while the homozygous recessive parent can only produce gametes with the recessive allele. We use a Punnett square to do the cross. The Punnett square will show probabilities of the genotypes of the offspring. This means that there is a measurable chance the cross will produce an offspring with a certain trait. And on the top, we will put the gametes from one parent, and we will put the gametes from the other parent on the side. Now look at the first square. We need to combine the gamete in the first column and the gamete in the first row. What is that combination? Yes, a dominant allele and a recessive allele. That makes a heterozygote. We always write the dominant allele first. Now how about the second square? Combine the second column gamete and the first row gamete. What do you get? Another heterozygote. Let's do the rest. All heterozygotes. Since one parent can only donate a dominant allele and the other can only donate a recessive allele, we can only get heterozygous offspring from this cross. We can express the results in several different ways. We can express the probability as a ratio. The genotypic ratio is 1 to 0 because all the offspring are heterozygous. There is no chance the offspring can be homozygous dominant or homozygous recessive. We can express the probability as a percentage. What is the chance the offspring will be heterozygous from this cross? 100%. What is the chance the offspring will be homozygous recessive or homozygous dominant? 0%. We can express the phenotypic probability as a ratio as well. The phenotypic probability is also 1 to 0 because all the offspring will have purple flowers. There is no chance the offspring will have white flowers. What is the chance the offspring will have purple flowers from this cross? 100%. What is the chance the offspring will have white flowers? 0%. So what will happen if we cross the F1 generation? Since the offspring are heterozygotes, they can produce gametes with either a dominant allele or a recessive allele. Let's do a Punnett square to find out the probabilities of the offspring. Gametes from one parent on the top and the other on the side. Okay, now fill it in. 2 dominant alleles, a dominant and recessive allele, a dominant and recessive allele, and 2 recessive alleles. So we have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive offspring from our cross. So that would make our genotypic ratio for the first generation 1 to 2 to 1. 1 dominant homozygote to 2 heterozygotes to 1 recessive homozygote. And percentages, from this cross, what is the chance of the offspring being homozygous dominant? 25%. Heterozygous? 50%. Homozygous recessive? 25%. And the phenotypic ratio? That would be 3 to 1, since the dominant homozygotes and heterozygotes would display the dominant phenotype. And percentages? From this cross, what is the chance of the offspring being purple-flowered? 75%. White-flowered? 25%. A dihybrid cross examines two characteristics and is much more complicated. Let's look at a cross between an individual that is homozygous dominant for both characters and an individual that is homozygous recessive for both characters. We are looking at pea plants again, but not flower color. One characteristic is seed color, while the other is seed shape. Yellow is dominant over green, and round is dominant over wrinkled. Now we need to produce gametes. The gametes will contain one allele for each characteristic. So for the first parent, what kind of allele can be donated? Only the dominant allele. And what about seed shape? Only the dominant allele. So all the gametes produced will have a dominant allele for seed color and a dominant allele for seed shape. What about the other parent? The second parent can only give recessive alleles for both seed color and seed shape. So all the gametes from the second parent will contain one recessive allele and one dominant allele. Now when we do the Punnett square, we have to put one parent's gamete combination on the top and the other parent's gamete combination on the side. Notice that we cannot split up the seed color and shape alleles, as they are both in one gamete. Let's combine. The color gametes need to be grouped together, and the shape gametes need to be grouped together. So from this cross, this is the only probability. What is the genotype of the offspring? Yes, the offspring are heterozygous for seed color and seed shape. What about their phenotype? Yes, the offspring have yellow and round seeds. What happens if we cross our F1 generation in this dihybrid cross? This is a bit more complicated. The parents are heterozygous for both characteristics, so there will be more gamete combinations. To figure out the combinations, we combine the first allele for color and the first allele for shape. Then the first allele for color and the second allele for shape. The second allele for color and the first allele for shape. And the second allele for color and the second allele for shape. That's 4 different gamete combinations. And the other parent has the same combinations. Now we have to put those gametes in our Punnett square. How many squares do you think we will need? To determine this, you need to multiply the different number of gametes each parent can produce. 4 times 4 is 16. Let's put those gametes where they belong. Now, try to fill in the squares before I do. This shows the probability of our offspring for the F2 generation. Let's determine the phenotypes. How many do we have that would display the dominant trait for both seed color and shape? 9 would have yellow round seeds. And how many would display the dominant trait for color and the recessive trait for shape? 3 would have yellow wrinkled seeds. How many would display the recessive trait for seed color and the dominant trait for shape? 3 would have green round seeds. And how many would display the recessive trait for both seed color and shape? 1 would have green wrinkled seeds. And what would the phenotypic ratio of this F2 generation be? 9 to 3 to 3 to 1. And whenever we have a dihybrid cross and the P1 generation has one parent that is homozygous dominant for both characters, and the other parent is homozygous recessive for both characters, the F2 generation will always have this phenotypic ratio. How exactly do these allele combinations occur? Well, it all comes down to meiosis and those homologous chromosomes. This is a karyotype of human chromosomes. A picture is taken of the duplicated chromosomes during cell division to produce the karyotype. We can pair up most of the homologous chromosomes based on their length and centromere positions. If you will remember, one chromosome of the pair comes from mom, while the other comes from dad. The only pair that might be different are the sex chromosomes. This karyotype shows the individual is a male, as one chromosome is the X, and the other chromosome is the Y. That combination typically produces a male. But what occurred to produce this combination? Let's look at the principle of segregation. It states the homologous chromosomes must separate during meiosis I. If the homologous chromosomes have different alleles at a particular locus, these different alleles will be separated into different cells. Meiosis II will just separate the duplicated chromosomes. We end up with half the gametes having one allele type at the locus, and the other half having a different allele type. We also have to consider the law of independent assortment. This states that if genes are in different chromosomes, they will be independently assorted. Why? Well, remember chromosome shuffling? Due to the random way the homologous chromosomes align at the midplane during metaphase I, we could have this sequence, or this sequence. And because chromosomes containing different alleles at the same loci would be pulled to opposite poles, they would wind up in different cells at the end of meiosis, resulting in many different combinations of gametes that can be produced. But it can get even more complicated. But what about those sex chromosomes? The X and Y chromosomes for humans are different. Do they contain different genes? Different species may require different arrangements of sex chromosomes to determine male and female development. For example, humans are mammals, and mammals have X and Y sex chromosomes. An individual with two X chromosomes will develop as a female, and an individual with an X and Y will develop as a male. The other 22 pairs of chromosomes in humans are called autosomes. Because there are different genes on the X and Y chromosomes, the genes found on each are called sex-linked. The X chromosome has over 1,000 genes, and no zygote can survive development if it lacked an X chromosome. But the Y chromosome has about 80 genes, and these are specific to male development. In particular, the SRY gene helps to regulate other genes during development for males. The X and Y chromosomes only have a few genes in common, but they are able to synapse during meiosis. Because females have two X chromosomes, they can be homozygous or heterozygous for genes on the X chromosome. However, since males only have one X chromosome, they only have one allele for all the genes on the X chromosome, and they only have one Y chromosome. Due to this, males are hemizygous for genes on the X chromosome and the Y chromosome. Whichever allele they have is going to be expressed. This can be observed with the disorder colorblindness. The gene that expresses the production of the structures to view red and green colors is on the X chromosome. If this gene is defective, it will result in red-green colorblindness or the inability to distinguish these colors. Since females have two X chromosomes, they have two alleles for this gene. Both alleles must be recessive for a female to be red-green colorblind. But males are hemizygous for this gene, and whichever allele they have is expressed. If they have the recessive allele on their X chromosome, they will be red-green colorblind. This is true for all 100 plus genes on the X chromosome. Is one allele always going to be dominant for every character? Well, the traits we've looked at so far have been determined by complete dominance. Many alleles do not display this type of dominance, and we also see codominance and incomplete dominance. In codominance, more than one allele type can express itself at the same time. A good example is the ABO blood group in humans. The phenotypes display different glycoproteins on the membranes of red blood cells. These glycoproteins can have carbohydrates for type A, type B, and a type that lacks either. Genotypes with two alleles that produce the A carbohydrate will result in type A blood, and a genotype with one allele that produces the A carbohydrate and a second allele that produces neither type also results in type A blood. The same thing applies for type B blood. And a genotype with two alleles that produce neither carbohydrate, the recessive allele, results in type O blood. But we see codominance in type AB blood because both carbohydrates are expressed. This is because both alleles are dominant. Incomplete dominance is when the heterozygote does not display the dominant trait. Instead, an intermediate phenotype is observed. In morning glory plants, we see three different flower types: purple, lavender, and white. The purple phenotype is due to a genotype of two alleles that produce a purple pigment. The white phenotype is due to a genotype of two alleles that produce no pigment. The lack of pigment results in the white-colored flowers. And the heterozygote has lavender flowers. It has one allele producing purple pigment and one allele that does not produce pigment. With just one allele producing pigment, it is not enough to make the flowers bright purple. Instead, enough pigment is produced to make the flowers light purple, or lavender. Co- and incomplete dominance demonstrate the molecular basis of alleles. In reality, alleles do not interact. Dominant alleles produce some type of product. Recessive alleles are usually defective and do not produce a viable product. That is why the dominant allele can express itself over the recessive allele. But are all genes like those examined by Mendel? One characteristic is determined by one gene? No, in fact most characters are polygenic. This means that two or more genes interact to produce the characteristic. We see this in human height, skin color, eye color, most of the traits we are familiar with. On the other end is pleiotropy. This is when one gene may affect many different characteristics. We see this in the SRY gene on the Y chromosome. This one gene regulates many different factors that result in the development of male characteristics. When mutations occur, gene disorders can arise. Some disorders are due to one defective gene. Albinism is due to a mutation in the TYR gene that codes for melanin production. Melanin is the main pigment found in skin. Defective TRY genes do not produce melanin, and if an organism has two recessive copies of the gene, then the skin, hair, and eyes will lack this pigment. Sickle cell anemia is a disorder due to a mutation in the beta-globin gene. This gene produces hemoglobin, the protein that carries oxygen in our blood. A certain mutation will result in an incorrect shape for the hemoglobin molecule. This causes the red blood cell to form a sickle shape. Individuals with two recessive alleles have these sickle-shaped blood cells, resulting in low oxygen levels in the blood and poor circulation in capillaries.